Welcome to PharmTech's Pharma Fundamentals video series. Episodes in this educational series aim to provide knowledge in the basics needed for pharmaceutical development and manufacturing, such as chemistry and pharmacology, breaking things down to the basics to help those working in the industry to better perform their jobs. Be sure to check the episode webpage for more topic information and resources. We hope you enjoy the series. Welcome to this episode of PharmTech's Pharma Fundamental Series. I'm Susan Hegney, lead editor of PharmTech. In today's episode, guest host Siegfried Schmidt, Vice President Technical at Parexel and a member of PharmTech's editorial advisory board, talks with Laura Lotfi, Associate Director, Project Management, Digital Projects at Charles River, about virtual control groups and how they are being used in drug development as an alternative to animal testing. We hope you enjoy the discussion.
Hello, everyone. My name is Siegfried Schmidt. I'm Vice President Technical at Parexel, and I'm also a member of the editorial advisory board for PharmTech.
My name is Laura Lotfi. I'm a director of digital product management at Charles River. My background is a toxicologist, and I specialize in digital transformation within drug discovery and safety. I focus specifically on integrating advanced data platforms, innovative methodologies such as virtual control groups, and my work is very centered on improving how we generate insight, make decisions, and bring safer and more effective therapies to patients. So I live and operate in the intersection of science, data, and regulation. My hope is to translate emerging technologies into approaches that are not only scientifically robust, but also aligned with regulatory expectations. So in essence, I help bridge innovation with real-world application in a highly regulated environment.
Now, Laura, you gave a very interesting introduction. So it sounds most fascinating, cutting-edge science. And of course, there have been 2 new guidances being published on virtual control groups. So perhaps you could give the audience a bit of an introduction. What is it that we are talking about here?
Absolutely. So thank you for the question. Traditionally, animal control groups rely on live, untreated animals that are studied alongside the treatment group that is receiving the new compounds or the new test molecules to provide a baseline to compare. Virtual control groups, on the other hand, use existing historical control data that simulate that baseline through computational methods. The fundamental shift is from generating new control data prospectively from animals that are on the experiment to leveraging high-quality existing data that are retrospectively available. So the goal is to maintain the same level of scientific rigor while using more efficient approaches and less animals.
Now, of course, we are all interested in using fewer animals in the research space. So this sounds very interesting, and obviously, I believe it's already been used. So is this a general thing that we can do with any type of animal, with any type of data? I suppose there are some restrictions, some limitations at the moment.
Yes, definitely there's limitation to the virtual control groups, but let me maybe set the stage up to what are the benefits to using it in the first place. So one of the important benefits, like you highlighted, is definitely the reduction in the animal use that aligns with the 3 Rs that are replace, reduce, refine. And at the same time, it definitely reduces the study timeline, bring in more from a scientific perspective, an improved consistency and statistical power as you're drawing from a large aggregated data set rather than relying on a relatively small, sometimes variable control groups in individual studies. The limitation, however, that we definitely can see from the virtual control groups are the data itself. It's highly dependent on the quality, the completeness of the underlying infrastructure that may contain sometimes biases or gaps that can affect the reliability of those results. They may also be less suitable for certain scenarios. For example, like you mentioned, there's some studies that involve new biological mechanism or rare endpoints, maybe some species that are not very well understood, where the limited historical data are not quietly there yet. So there a lack of sufficient historical data that is available. In those cases, traditional approaches may still be necessary.
So obviously, as you mentioned, we need to have a good data set to start with. Now, if I'm a newcomer to this field, how would I know whether there is a data set that is sufficient, or maybe asked differently, who decides whether the data that are available are suitable, unbiased, etc.? So maybe you can explain a bit more about that.
Of course. So how the virtual control groups are created, developed, and kept to be, validated or in check is a very extensive methodology that relies on, first of all, like you mentioned, a very careful, curated historical control data collected from previous study. That data has to be standardized, harmonized to ensure the consistency across different sources. From there, we developed statistical methods like machine learning methods that are applied, combined with biological and toxicological expertise from various subject matter experts to match the characteristic of the treatment group that are as close as possible. So the matching criteria to the experiment are crucial. This is a key step that includes aligning study conditions, matching relevant attributes, ensuring the final model reflects established scientific best practices. And it relies heavily on something that we describe to be the validation or the experimental of the scientific approaches. So typically, the validation of a new scientific methods rely on using this new technique in a retrospective manner and try to reproduce the outcomes using the new methodology. So this typically for the virtual control groups helps demonstrate that this approach is reliable and comparable to the traditional methods that is using the concurrent control group. In addition, the validation definitely requires strong statistical rigor, predefined acceptance criteria that are in collaboration with the experts like toxicologists, pathologists, and also the alignment with the regulatory expectations. This level often depends on specific context of use, which the virtual control groups will be applied into, and not just a one-fit solution for multiple study designs.
So, if I understand this all correctly, only few companies, people are already developing these virtual control groups. It's not something that every R&D company is developing because it seems it's very sophisticated and requires a lot of understanding of various subjects and expertise in a lot of areas. So is this something that is already widely applied, widely developed, or is this in the very early stages? Where are we?
I would say it will depend on who you ask. Definitely, with any new technology, there is a more mature companies versus how they experiment versus something that is innovative for new companies that are adopting this. I would say in general, most companies begin with a hybrid or prospective approach to understand what this new technology is for them. So we are seeing a new pattern where we are using the virtual control groups alongside with traditional animal controls. And I would say would be like early to mid on the adoption curve, because now we've shown scientific validity throughout multiple scientific publication consortium, but we want to see it in the practice. So moving from the the theory into practice, how that translate. So this methodology allows companies to build confidence, generate comparative evidence, demonstrate that this new approach produce equivalent result. I would say with this approach, the main challenges include ensuring that the high quality data and standardized data is being shared with the community, gaining regulatory acceptance, building models that are transparent, reproducible in the scientific field, and fit for the intent of purpose is definitely what we need to keep focusing on, working together as a community on. I would say also from a regulatory standpoint, it's critical to clearly define how the virtual control groups and where they will be used to demonstrate that acceptance and meets the necessary scientific compliance. So I would say from that adoption curve, we are early to mid, but also on the technical side and the understanding of the scientific, we are definitely of a more mature type of conversation. So the question is now becoming not if we can use virtual control groups, but how to best and where to best use it.
Now, as you say, we need to understand how, and this how, of course, also requires approval by the regulators. We know we have these guidance documents just been published, but in general, would you say that regulators are very keen on this, are encouraging it, and so at what level is their expertise in general there? Because, as you say, this is not something that is mainstream yet.
So new alternative methods, I believe, from the regulatory perspective, has been there for a long time. I do think that in the recent years, we are seeing a shift in that landscape to be a little bit more tactical, more applicable, more, I would say, encouraging to generate those new alternative methods. What we're seeing today with the European Medicine Agency or EMA new guidance or opinion have been always generally more proactive in the encouraging of the alternative methods to animal testing. However, today, this is a strong institutional support for data-driven and innovative approach that we are definitely seeing, including virtual control groups on their roadmap and in their support to the Victor Consortium. In the United States, the FDA also is moving in this direction, I would say, with their FDA amendment number 2, with the new big push around the new alternative methods. They were typically with a more case-by-case approach with the iStand program. However, currently we are seeing a strong emphasis on the need of new alternative methods and on its validation. So they're pushing towards clear context of use, ensuring that any new methods meet the established standard for reliability and the safety of the patients. So I would say there's definitely a shift toward that on multiple regulatory agency scales in a different style, perhaps, but definitely on the same direction.
So do you know, Laura, if any of these virtual models have been already presented to the regulators for consideration or been discussed in early meetings or something like this?
Well, as you can see with the EMA, the qualification opinion was based on data they reviewed, right? So definitely they have seen that. They are very proactively engaged and participating in driving the future context of use and how that could be defined. So the collaborative space is definitely created there. We've heard from the FDA as well, their openness toward the new alternative methods review, whether with the iStand or whether in different collaboration spaces. So the data has been presented to them in one context or the other. And definitely we're seeing openness to collaborate further on how we can include those in submissions for regulatory review.
Well, it all sounds very promising, Laura. Do you, of course, the one benefit is we don't use live animals, which is Definitely worthwhile. But from a cost perspective, do we look at something similar in cost, or are you aware of any comparison there?
When we speak about any shift in the study design, definitely in general, there will be a cost impact to be revised and, in discussion with the clients and the regulators, how this could be incorporated. So as we understand how to implement the virtual control group, what are the context of use for it, we are looking to see how that impacts the whole study and program design, that it is the timeline, the cost, the impact on the endpoints that are included, how we can best leverage this technology to make sure that we are working efficiently, but also making sure that our scientific rigor is kept to its utmost high level or high quality.
That is excellent. Well, Laura, I don't have any further questions. Anything you want to say to conclude our talk today?
Sure. So I'm very excited to see, the virtual control group becoming a topic that everybody is interested to learning more about, to see how it can be incorporated to help drive the 3 Rs, but also to help include new alternative methods further and validate them. So I invite people to be open-minded to learn about this new scientifically method and to keep challenging a status quo experimental design when it is actually feasible, and how to best keep our patient at the forefront of our innovation.
That's wonderful. Thank you very much, Laura.
Thank
And
you.
hope to see you again. Bye.
Thank you.
Thank you for joining us for PharmTech's Pharma Fundamentals video series. If you would like to suggest a topic for discussion or participate in an episode, please email us at ptprojects.com and include Pharma Fundamentals in the subject line. We look forward to you joining us next time.